Okay, previously, we've talked about sickle cell anemia, which is an inherited disease caused by mutations in the DNA. And the gene for that was cloned and sequenced a lot earlier than for this next disease we're talking about, cystic fibrosis. Now, with sickle cell anemia, it was fairly obvious that the problem with the hemoglobin protein, and particularly the beta chain, so scientists knew where to look. They knew they had to clone the hemoglobin genes. With cystic fibrosis, it's not so obvious what's going on there. It's not obvious which protein or which product was at fault. All that was known was, well, cystic fibrosis, if a person suffers from the disease, they get very thick, sticky mucus in their lungs, which causes them to get repeated infections, and this leads to lung damage, and their life expectancy is a lot lower than healthy people. And it was new, it was inherited in a recessive fashion. They could tell that some people were carriers. Okay, so if you were if you had 2 copies of the normal gene and you were healthy, if you were heterozygous, there were some people who obviously were carrying a gene and passing it on to their offspring, but they were healthy themselves. And then you needed 2 bad copies of the gene to have cystic fibrosis. The trouble is, the pattern of inheritance was very clear, but no one had any idea what the gene was. Right, now, in order to find out whereabouts in the human genome it was, what they had to do was they had to make something called a genetic map, which we'll just explain down here. Right, so what I've got here is a pair of homologous chromosomes, okay? And we're looking at two gene loci here, right? We've got this one, which is A, or it can be little a, and the next one a bit lower down on the arm of the chromosome, the 2 alleles it can have is big B or little b. Okay, so I've drawn the black chromosome there with large A, large B, and the red one with little a, little b. Now, if chromosomes, because they're on the same chromosome, we could say that those 2 loci, A and B, are linked. If no genetic crossover occurred, you can see that the only possible genotypes you could have would be big A, big B, and little a, little b. It would be impossible to have, that or that. That would be impossible, those 2. Of course, they are possible because, okay, they're on the same chromosome, but you get material which is passed over between chromosomes. Remember, it's called crossover. It's recombination of DNA molecules, and it happens at meiosis, and it's one of the main drivers for genetic diversity. Okay, so if you have a look at, well, there's been a crossover event here now, and that means we can have the 2 phenotypes, little a and big B, and if it happened in a slightly different way, you could end up with that. Okay, so because those genes are quite a long way away from the chromosome, we say that they're not tightly linked. Okay, let's imagine if you had 2 genes, and let's do that. So, like a red gene there and a blue gene on this chromosome. They're very close together, and so they would be very tightly linked. Okay, you would nearly always get them. They'd been inherited together. So whatever the gene is, they wouldn't tend to separate from each other, and that's because they're really close together. Whereas if you had a third one, let's say if you had that yellow one, that's a bit further away. So that would be, with the red one, that would be inherited together usually, but every now and again you would get a recombination event, and so you can tell it's close but not so close. So this idea of seeing how far away the genes from each other by how often they undergo recombination events is called a genetic map. Okay, so now the first job was to find out which chromosome the cystic fibrosis gene was on, and it appeared that, well, it was actually chromosome 7. And it took quite a few years to find out which chromosome it was, but they decided it was chromosome 7. So then made a detailed genetic map, lots of different genes, and find out how far away they all were from each other. So there's maybe they use 100 different markers over the chromosome. So, you know, if I just choose that one at random and that one, they're not going to be very tightly linked because they're really far apart. Okay, but they're going to be more tightly linked than that one and this one here, because that's even further apart. So they constructed a genetic map, and they knew where the cystic fibrosis locus was. They didn't know what it was, but they knew that it always seemed to be inherited with certain other genes. They looked at cystic fibrosis families. So they found out genes that were very tightly linked to it. So let's say they found that red gene there, that was very tightly linked. And the one that they found, it had the interesting name of DOCRI917. Now, it doesn't code for anything interesting like eye color or anything like that. It was just simply a sequence, it's a DNA sequence, and it didn't code for any protein, probably because most DNA sequences don't code for proteins. But they knew it was there, and they could see with cystic fibrosis patients, if you had the mutation, you always had a particular version of this gene here, and they knew where that was on the chromosome. And they knew, therefore, that the cystic fibrosis gene, I'll do that in blue, must be really, really close to that. So they knew which bit of chromosome 7 that they had to sequence. At this stage, they couldn't have sequenced the whole of the chromosome. It was in the 80s, it was before the Human Genome Project, before it was automated. Sequencing was a very laborious process, so they couldn't do the whole chromosome. They had to just zoom in on this bit they were interested in. Even so, even though they knew the bit, it still took quite a long time to find the candidate gene. Now, how do they find the candidate gene? Well, they were looking in the sequence for something called an open reading frame. We know that probably only 3% or something of human DNA actually codes for proteins. The rest of it, I'm not entirely sure what the rest of it does, but it does have some function. But open reading frames, you can tell because you will see they'll have a start codon. Telling which is the AUG codon, right? Well, it's in DNA ATG, start codon, and then you'll have a whole load of the sequence, and then you'll have a stop codon. So they identified an open reading frame sequence, and they found out that it coded for a protein. There was a lot of exons in there. There's 27 exons. It was split up into 27 exons, so 27 introns between them. It coded for about 1,400 amino acids. And they set about trying to find the function of that gene. What did it do? Well, the obvious thing to do is find the amino acid sequence. And very often from the amino acid sequence, you get things called motifs, where you'll get certain amino acids always occurring together. You know that's going to form an alpha helix. Certain amino acids always going together. That's an ATP binding site. Little clues to what the protein does. And eventually they found that, and then they purified the protein as well, obviously when they knew they were looking for. The protein is given the name CFTR, which is Cystic Fibrosis Transmembrane, transmembrane regulator. That's the name of it. So it's a transmembrane protein, as the name suggests. So I'll draw that in there. There's a lipid bilayer, and there's our transmembrane protein. Right, what does it do? Well, it acts as a channel for chloride. So this is inside the cell here. It's the cytoplasm. It acts as a channel for chloride ions, and chloride ions can pass through there and leave the cell. Now, how does that lead to sticky mucus? Well, if the chloride ions stay in the cell and they don't get outside, that will tend to, water will this lowers the osmotic potential, obviously, and draws water out of the cell into the outside where the mucus is. This is going to be all the mucus and the lung cells. And if there's not enough water in that mucus, the mucus is too thick, and that's where all the problems come from. So there we've got our cystic fibrosis protein. That's the normal protein. Well, what has gone wrong in cystic fibrosis? Well, in most cases, I think it's about 30% of cases, what happens is there is a deletion. It's called the delta 708 mutation, because, or 508, sorry, because there's amino acid at position 508 has been deleted. The amino acid is phenylalanine, and that's missing from the protein. Right, so that means there must have been 3 bases deleted here, and it leads to an amino acid missing from the protein. Now, quite often, if you're missing an amino acid from a protein, it might not make any effect at all on the protein's function, or very, very limited. But this one does have a large effect. So what it stops happening is the protein, it can't assume its correct 3-dimensional cell shape. And it's non-functional. It can't let chloride ions pass through it. Now, there has recently been a drug available to treat this. It's a really expensive drug, and there's a bit of controversy because it was so expensive. And what it's called, lumacaftor. And the way it does is it binds to the ribosomes, and it sort of helps that protein fold in a slightly better way than it would do otherwise. It's not as good as the healthy version of the protein, but it does work a bit, and it can let some chloride ions through, so it works that way. Now, that was the first mutation that they discovered, and this is the most common mutation, this delta 508. But cystic fibrosis is quite a wide range of mutations. There's over 1,000 mutations have been, greater than 1,000 mutations have been identified, different ones. Okay, and some of them, they all do slightly different things. So some of them, you get a stop codon in the middle of the protein. So you get a mutation occurring where it should be a coding for amino acid. You get one base change, it makes it a stop codon. So you end up with only a partial protein, a truncated protein, and that's not going to work. Other mutations are in the splicing regions. Now, we said that this has actually 27 exons. I'm not going to draw 27, I'll just draw a few. There's the exons, and it's got introns in between them. And what's going to happen is those green introns, bits of RNA, they have got to be looped and spliced out. They go like this, okay? Before the mRNA is, so they get spliced out and thrown away before the mRNA is translated. If you have a mutation in the region here, just before, there's a sequence there which is needed in order for the mRNA to splice correctly, and if it's not there, then that won't happen, and so you don't get a functional protein. Now, it seems strange that this one gene, there should be so many different mutations, and the fact that these mutations are obviously persisting and they haven't been removed by evolution. And there is a suggestion, like with sickle cell, that maybe being a carrier is maybe some evolutionary benefit. Just from the mere prevalence of the mutations. And there has been suggestions, although I don't think they've been confirmed or proven, hypothesis that it may give some mutations in the CFTR gene might give some immunity to, or resistance to cholera and disease where you get like loads of water leaving the body and diarrhea. It may be that it has some kind of protective effect in that disease when you only have one copy of the mutation. So that's cystic fibrosis, then a lot more of a difficult problem to sort out than the sickle cell, mainly because they were trying to find a needle in a haystack, but they didn't know what they were actually looking for.